Hello students, today we are discussing continuation of the development of embryo, that is a monocot embryo. Already we studied about dicot embryo. In that dicot embryo, normally the plummules which one can be present in a distal part and cotyledons are present lateral parts and whereas in monocot embryo, in monocot embryo, the main differentiation, it will be present the terminal cotyledon. Here cotyledon we called as scutellum. Then lateral plummule, that is main difference between dicot and monocot variation and all. Here there is no globular, that heart shape, unlike dicot and all. Here normally the development one can observable in monocot and all. That particular developmentation, it will be present like without any variation, without any globular one, like continuation, the development it will be present under, then like that cells which one can be formed under, but finally, and very important point of view diagrammatically, which one can observable, this is, that is a suspensor. Then below that suspensor, already we know, below the suspensor is normally it will be present into the radical. Radical it will be present in that monocot. This is only very important. In monocot, the lateral plummule part will be present. The terminal part. This is the terminal part. The terminal part, that is cotyledon. Here cotyledon, otherwise we called as scutellum. Normally it will be covered with sheath and all, that scutellum and all. Then related to grass, that continuation and all, A, B, C, D, like that to growth and development and all. Then which one can, one among these, that grass, if you observable, in that grass, that after complete maturation of that particular embryo, if you observe, that is a rough diagram related to for understanding purpose, but here very important, related to that coleorhiza, then root cap, then here that is shoot apex. This that coleoptile. This is we called as scutellum. This is epiblast. So in our textbook also they have given, apart from this one, a important one, in epiblast, what is the function of epiblast? It helps differentiation of germ layers. Apart from this one, in monocot, generally one can observe coleorhiza, which one can covered with root cap, as well as related to coleoptile, related to shoot apex and all. In that dicot, one can observe that is epicotyle, hypocotyle. But here coleorhiza as well as coleoptile. That is major differentiation as well as scutellum, as well as formation of the particular plummule and all. This particular diagrammatic view, one can representation of the grass and all. In our textbook they have given LS of the maize. There the scutellum, which one can be observable, LS of the maize, which one can be observable. This is the coleorhiza, coleoptile. This is endosperm. This is we called as scutellum. This is LS of that maize plant and that is, this is the pericarp. Pericarp, after fertilization, after completion of this one, the ovary membrane, it converts into the pericarp and all. This is the maize, this is the grass, LS of that particular grass and all. But here one can observable in monocot and dicot, some of these particular plants, normally elongated coleoptiles it will be observable. Some of these dicot embryos, elongated, curved, that coleoptiles, that is nothing but related to cotyledons, one can be observable in dicots and all. That is capsella. In Capsella bursa pastorica, such type of these plants, one can observable elongated, that particular coleop related to cotyledons, one can observe in elongated, that curved, which one can be present, that cotyledons. Then in Capsella one can observe. Normally it will be present lateral and smooth under. There it will be some exception. Some of the plants which one can be observable, normally in dicot 2 cotyledons, in monocot single cotyledon present. But some of the plants which one can be present, more than 2 cotyledons are present. So normally in that Loranthus, especially in monocot, it will be present only one, but here 2 to 6 cotyledons present. In Loranthus, it will be present 2 to 6 cotyledons. But some of the plants, cotyledons completely absent. Suppose in Cuscuta, because of parasitic nature, the cotyledons completely absent. In Cuscuta, the cotyledons are absent in Cuscuta. Like that, these are some exception cases which one can be present and all. But apart from this one, some other organisms like Orobanche, as well as Orchidaceae, as well as Trichileria, these different types of the organisms, their plumule, cotyledon, as well as that particular parts which one can be, may not be identified very clearly. That a plumule, that a cotyledon, as well as a radicle, these parts may not be identified clearly before shedding of the particular seeds and all. In that Orobanche, that is one type of that weed plants and all. Then Orchidaceae, then Trichileria, these 3 different types of this one, that plumule, cotyledon, this particular radicle may not be identified very clear, may not be formation very clearly. Then you say, what is the first organ one can develop while growth and development of the organogenesis in that embryo and all. Normally in 16 to 32 cell stage, the organs which one can be formed, ready to start and all. What is the first organ? Normally one can form into the cotyledons. If it is monocot, that we call as scutellum. If it is dicot, the name one can represent it as this one, cotyledon. The first organogenesis in that embryonic development and all, first organogenesis, that is scutellum or cotyledon, it will be present and all. Like this. In this, that is in monocot as well as dicot embryos. Then apart from this one, actually in our book they have given last one, polyembryon and all. To avoid that confusion, here only one can read, then to avoid the easily one can remembering and all. So what is meant by embryo? Where one can be formed embryo, one male gamete, one can fusion with the female gamete, then it forms into the zygote, then multiple division, one can formation of embryo, that is a common. Then if it is formed more than one embryo in embryo sac, more than one embryo, that is extra embryo, the phenomenon which one can develop more than one embryo in one that particular ovule in that embryo sac, that is that condition we called as polyembryony. embryony. Then, what is the main adaptation of this polyembryony? Why one can be formed? Regarding related to cell division, polyembryony one can take place. Normally, it is the parasitic adaptation. Who is the scientist? First one can discover about this polyembryony. That is Leeuwenhoek. Leeuwenhoek scientist identified this polyembryony. It is the main important characteristic features of the parasitic adaptation. Leeuwenhoek scientist one can be identified. That Leeuwenhoek. Then what type of plants? Very commonly one can observable polyembryony. They must give different options, either angiosperms, either pteridophytes, either gymnosperms, either thallophytes, like this one can give. Then your answer will be the gymnosperms. So most commonly one can formation of polyembryony, that is in gymnosperms. So commonly observed in gymnosperms. Which one can observe in gymnosperms? Then what one can arise this? What is the main reason why it will be formed such type of this polyembryony and all? So what can arise is that polyembryony. The polyembryony, which one can cause this again, whether it is a true, whether it is a false. There are two types of polyembryony. Other than gymnosperms, in angiosperms also it will be possible. Different causes and what are these? So what can be that arises? So polyembryony. So how one can be rises in that polyembryony related to in gymnosperms it is common. What is the commonly it will be present related to in this particular commonly which one can be represented either multiple formation of embryo sacs or within ovule embryo sac it will be present. Normal embryo one can form into the egg cell under or oospore it will be formed under. Other than oospore, if it is fusion of male gamete into the either synergies or antipodals or integumentary cells or nucellular cells, like that, it will be possibility of this one. In this one, when it is called as that particular that false polyembryony. False polyembryony in that sense, multiple embryo sacs, if it is present in one ovule, then if it is fusion, that is we called as false polyembryony. Normally, that multiple embryo sacs present in ovule, one single ovule, multiple one, that is we called as false, that particular this one. Normally one can observable to the fragaria, most common example, that is the in fragaria plants, it normally one can observable. Then apart from this one, then the true polyembryony. True polyembryony means normal embryo where one can develop inside the ovule in that respectable area. If other than that extra embryos are formed, that is we called as that true polyembryony. True polyembryony, the extra embryos which one can develop either embryo sac, either synergies, either antipodals, either integument. That means within a ovule, if it is fusion of multiple that particular pollen grains, one can fusion. That means male gametes one can fusion. That is we called as true polyembryony. That means either embryo sac in that within that embryo sac, if it is present in synergies or antipodals or integuments or nucellus, any one among these one. That means within the ovule, if it is takes place within the ovule, then that is regarded as true polyembryony. Then in this true polyembryony or false one, normally how one can causes either these two different types of this one, either one among this one, fragaria, this one. Suppose if it is formed multiple embryo sacs, normally why one can causes multiple embryo sacs under normal megaspore mother cell after meiotic cell division, there will be formed 4 haploid megaspores are present. In that 4 haploid megaspores, 3 will be degenerate, 1 will be functional megaspore and all. But in case all will be functional, then it will be present to 4 embryo sacs are present. Then in that particular cases, rare conditions, it will be present in that false, it will be regarding related to one can be takes place and all. That is such type of development of polyembryony that regarding related to this one, in that false one, that is we called as regarding related to mixed or simply within that same, it will be called as very simple polyembryony. The false polyembryony, otherwise we call it simple. That means multiple embryo sacs, which one can involve. That is, we call it a simple polyembryony. Other than this one, like brassica, in brassica plants also one can be observable. In that particular true polyembryony, we also observable related to that particular things. Either multiple pollen tubes enter into that particular, that means mixed polyembryony. It will be formed into the either mixed polyembryony. In that mixed polyembryony, that means multiple pollen tubes. Whenever one can enter into the multiple pollen tubes, then multiple pollen tubes, if it enter and fusion of either syringes or antipodals or regarding related to integumentary cells or nucellus, that is which one can represent, that is mixed polyembryony, we call this. This mixed polyembryony commonly one can observable into the allium. In allium plant as well as in that almus plant as well as plantago, such type of these plants normally one can observable, that is mixed polyembryony and all. Then apart from this one, either embryonic cleavage, normal after fusion of male and female gamete, normal it will be divides into the mitotically and develops into the, if it is dicot, globular, if it is monocot, normally one can form. If it is zygote, 2 embryos are formed. 2 embryos differently one can be developed. That is embryonic cleavage. Either embryonic, that cleavage. In that manner also polyembryony, it must be possible and all. In that one, in that embryonic cleavage, normally one can observable in that nymphia as well as orchidaceae, such type of that particular plants one can be observable. Either nymphia as well as orchidaceae and tobacco plants, normally one can observable embryonic cleavage. That means instead of one, multiple, either 2 or 3 or 4, which one can separately within that ovule, it will be developed. That is we called as that embryonic cleavage, polyembryony, normally one can observable. That examples are important here. Then apart from this one, another very important, multiple number of times in exam, in that previous NEET, APMT and CET and all, that exams which one can be asked and all, that common, very commonly which one can observable, that is nucellar polyembryony. Nucellar polyembryony or sporogenic cells development of polyembryony, otherwise it is called as sporogenic cells polyembryony and all. Then nucellar, that polyembryony, commonly one can observe, normally nucellar cells it will be present diploid one, the embryonic which one can develop into the diploid condition only, multiply it will be present. It is normally one can observable into the citrus plants as well as mango plants as well as opuntia, there one can be observable. More commonly citrus as well as opuntia as well as mango, nucellar polyembryony or sporogenic cell, that polyembryony normally one can observable these type of these organisms and all. It is many times in exam point of view they ask and all. Citrus, nucellular polyembryony commonly one can observable any one among the example. That is rarely one can ask, but for understanding purpose one can listen. Then apart from this one, suppose in that allium, how many number of polyembryos which one can formed in allium? 5. Suppose normally that in allium sepa it will be present 5, it will be present. One is zygote, then another one is that synergis, another 2 are antipodals, one is that particular that integuments. Like this, which one can be present? 5 embryos are present. So in that citrus 2 to 4 polyembryos it will be present and all. Allium that 5 it will be present and all. That is depends upon type, that cleavage, which one can more either false or true, that particular which one can be mentioned and all. These embryos, after formation of these particular embryos and all, then the seed which one can be formed and all. After completion of the fertilization, then what is the steps which one can be involved and all. Commonly in that particular flower, that calyx, corolla, it will be drooping off, style, stigma, it is also drooping off and all. Inside that particular, after completion of this particular embryo as well as placenta, in that post fertilization events and all, here in that post fertilization, in that event, that ovule it converts into that particular fruit, then ovary membrane, that is it will be formed into the pericarp, then integuments of ovule, it will be present, that is that seed coat it will be formed. Then ovule it converts into the seed, then ovary it converts into the fruit, then integument, then the funicle of this ovule, funicle of ovule, that is the stalk of seed, then synergis as well as antipodals, then synergis and antipodals are, that is that drooping off. Then if it is present nucellus, if it is persistent, if nucellus will be persistent, then that is we call it as perispome, nucellus, which one can be present? Micropyle. In that micropyle, one can observable like micropyle of the seed, micropyle as it is, micropyle of the ovule, micropyle of the seed. So in that particular after fertilization, what is this one? Match the following compulsory, they must be asked. One can remember carefully. Then apart from this one, these particular seeds, then after formation of this one, that fruit, how many types of that fruits, which one can be presented on? Fruits. In that fruits, different types of that fruit, seeds, everything one can study it in that first year and all. Then here, according to that basic concept and all, there are either fleshy fruits, either dry fruits. Fleshy fruits like that particular guava, orange, such type of mango, it will be present and all. Some of these are dry like groundnut, it will be present and all. Apart from this one, some of these one true fruits, some of the fruits are false fruits are present. The fruit which one can develop from ovary, that is we call it as a true fruits and all, like mango, like orange, all these fruits and all. But the fruits one can develop other than ovary, that means thalamus. That is such type of fruits we call as false fruits and all. False fruit. that false fruit normally one can observe like apple as well as that strawberry, which one can be observable under. Rest of most of the plants one can observable, that is the true only. But in that particular fruits, inside that fruit, multiple number of seeds. Some of these fruits one can present a single seed, it will be present. Then suppose in cereals, most of these one, it will be present single seed, it will be present under. But suppose in mango, except in polyembryony, normally one can observable one fruit present, only one seed only it will be observable under, either beans, everything under. Then if it is present, suppose in ficus, how many number of seeds one can observable throughout the ficus plant under? Number of seeds. Normally one can observable 850 billions of seeds present. In ficus tree, one can observable 800 billions of seeds in a plant. In one particular plant, that many number of seeds. Suppose orchids are present. In orchids, several thousands are present. Orobanchi, striga, such type of these are several thousand is present. In normal one or two, these are some exception. They also one can give one particular questioning under. In that questioning under, one can remember very carefully under. Then some of these one, after fertilization, some of these parts, it will be persistent. Suppose calyx persistent like brinjal as well as tomato, it will be persistent. Some of these one can formation of this one, aerial part. Some of these one can formation of this one, carancul under, related to that aerial, like lychee. Then carancul, like a castor. So after fertilization, which one can helpful for seed dispersal, easily dispersal of that particular seeds and all, which one can be supporting. Then some of these fruits one can develop without fertilization and all. The fruits one can develop without fertilization. Then what we call the development of the fruits without fertilization, parthenocarpy. Without fertilization development of fruits we called as parthenocarpy. The best example one can observable into the banana, that is the seedless one. Parthenocarpy nothing but one can induce artificially also. The parthenocarpic, either apple, either grapes, artificially one can induce this, which one can development of this one. Then what we call as development of the seeds without fertilization, that is we called as apogamy. There is also number of times one can ask, what is it by apogamy? Apogamy. That last subtopic, here one can given that partheno, that is polyembryony as well as apogamy. That is only one can given. Apogamy means without formation of seeds, without fertilization. That is formation of seeds, that is without fertilization. We call it as apogamy. Normally one can observe almost all type of the grasses as well as asteraceae. Such type of these plants one can observe apogamy, like asteraceae as well as in the grasses. Normally, what is the importance, what is the use, what is the benefit of this apogamy, apomixis and all, which one can develop hybrid varieties of the seeds, in that farmers within short span, more quantity, quality, such type of this particular yielding of the seeds, which one can be produced and all. So it is economically more beneficial related to this apomictic particular type of that particular seeds and all. Then apart from this one, some of these seeds immediate after formation, which one cannot able to germinate and all, then it will be alive. Then that particular period of time before germination, that particular period of time we call as dormancy and all. That seed dormancy. That means we call as viability of the seeds. Different types of seeds, different viability natures one can be present and all. Different viability natures are present. Some of the seeds like rice, wheat, all this one, within few minutes that seed viability it will be completed and all. Some of the seeds one can be present maximum period of time. that seed viability. Viability in that sense, it will be alive. Still, it will be alive until particular suitable condition, environmental condition, once one can ready to germinate and all. That is seed viability. That is in that seed viability. In that this particular seed viability, for example, lower grade like cereals and all, rice wheat and all, which one can few minutes, like oxalis seeds. In oxalis seeds, only few weeks, less than 1 month or 1.5 months like this, only few weeks, number of weeks only it will be present. Then recently one can found that is Phoenix dactylifera in Dead Sea region, that is the number of years, that particular viability of the seed, which one can be present, that is we called as date palm and all. Phoenix dactylifera, date palm, or Phoenix that dactylifera, that is a date palm. It will be present 2000 years, nearly. Recently one can observe this one in Dead Sea, in King's Herod region, which one can be identified. Then apart from this one, lupinous seeds, it is also one type of this Fabaceae family under Lupinaceae and lupinous under in that Fabaceae. It will be present nearly 10,000 years. Lupinous arctica. In Arctic tundra region, normally one can observe these seeds, Arctic tundra region, then lupinous arctica, that viability of these particular seeds are 10,000 years, maximum 10,000 years. Then this lotus, what type of this viability of these lotus seeds and in that lotus seeds. In that lotus seeds, normally one can observe in 400 to 2000 years. It will be present 400 to 2000 years. Lotus seeds and all. Like this, different types of this viability of that particular seeds and then outer seed membrane, whenever favorable conditions, which one can ready to germinate and all. Apart from this one, artificial hybridization also act as a very crucial role under artificial hybridization instead of natural that pollinating agents, which one can formation of embryo as well as endosperm, all these aspects under. Then normally one can observe, that means emasculation, which one can remove of these anthers if it is to avoid that self pollination and all, or bagging in that sense, one can cover stigma of that particular flower if it is a bisexual one. Unisexual, no need emasculation bagging. Normally in bisexual, which one can, that particular devices, outbreeding devices already we discussed, that devices naturally may not be available artificially, which one can increasing that hybridization of these plants and hybridization, that means cross hybridization, that technique normally one can use emasculation as well as bagging, which one can cover to avoid to entering of within the same pollen grains of the same flower, to avoid that self-pollination and all. That is very important aspects and all. So all these particular concepts, once again go through, one can be read. Apart from this one, in this particular chapter, what is, apart from this one, until last couple of years, they did not ask diagrammatically and all. There will be chance this year, one can be asked and all. Whatever the textbook diagrams, that much be enough. That is the apple, which one can be given onion seeds as well as beans. It will be given in that, especially in that beans, whenever they are given the dicots and all, there. This is the, that is the plumule, this is the radicle. These are cotyledons. This is the beans. In that beans, if it is, which one can LS section of that beans and all. If it is normal section view, one can observe it is the hilum. Then it is the micropyle, micropylar region. That hilum region, it will be present, hypocotyle, which one can be present. Then apart from this one, castor, this also one can be given. In castor, once one can observable, that is the endosperm, that is the pericarp, that is the hypocotyle, this is the caruncle part. Like this, which one can be given, one can go through that particular diagrammatic, whatever other than this textbook I have given here. Remaining as usual one can be present. Then in that in the grass, whatever LS section of that embryo also, that is also important, many times they ask. In that previous years, multiple number of time they asked in that exam point of view, that is normally one can observable in geitonogamy, as well as apart from this one in polyembryony, especially nucellular polyembryony, where one can be observable. Then apart from this one, types of endosperms. Then another very important, many number of times one can ask the function of tapetum. Then how many functional megaspores normally one can observable in that normal condition. If it is polyembryonic condition, more than one. Normally only one functional megaspore it will be present. In coming to this one, in that pollen grain, in pollen sacs, in anther, especially tapetum and all, in megaspore, in that apart from this one, ploidy, that is also number of times one can ask and all. That ploidy of that particular polygonum type of embryo sac, what is the ploidy and all? Many number of times one can ask, what is the name of that female gametophyte? Mention the name. Another, many times one can ask, within the same plant, the transfer of the pollen grains, one flower to another flower of the same plant. Then what we call such type of the pollination and all. That is commonly we call as geitonogamy. Then ploidy of this number of this one. Then apart from this one, then what is the difference between parthenocarpy, parthenogamy, apogamy, as well as polyembryony, especially nucellular polyembryony. That is, in these particular areas, many number of times. Apart from this one, remaining concepts also act as a very important role and all. Like that, once again, once or twice, which one can go through and all. In that any other things, once again, one can be recapitulate. Okay, thank you.